Everyone. Today we will discuss the topic organelle genomes. This topic is from the module 1 and this is the part 1 of the organelle genomes. The genome present in the chloroplast and the mitochondria are called the organelle genomes. The mitochondria of all the eukaryotic cells and the chloroplast of the plant cells are the only organelles that have the different types of the genomes. That's why we call this the organelle genomes. Mitochondria and the chloroplast occur and present outside the nucleus in the cytoplasmic genes and also known as the organelle genes or the extracellular genes. The replications and the mode of inheritance of the organelle genomes are totally different from the genomes which is present in the nucleus. These mitochondrial genomes are totally different from the nuclear genomes in the size, and these are the different among the different species to species and from one order to another order. The animal cells have the genomes of approximately 16 kilobase pair and represent the smallest mitochondrial genomes in the eukaryotes. The yeast possesses a much larger genomes. The yeast is a eukaryotic cells and varies among the different strains, but the total molecular weight of the mitochondrial genomes in case of the yeast is about 80 kilobase pair. And it makes about the 18% part of the total DNA which is present in the yeast. Apart from this, the plant mitochondrial genomes are the largest type of the mitochondrial DNA which is present in the eukaryotes, and it is the more complex than the animals. Now we will discuss about the mitochondrial DNA, and it is represented as the mtDNA or the mDNA. The mitochondrial DNA, it is the DNA which is present and located in the mitochondria. And you know that the mitochondria is a cellular organelles and present in the eukaryotic cells, and its function is to convert the chemical energy from the food into the form which can be acquired or can be utilized by the cells for their purpose. And this energy is generally known as the ATP, adenosine triphosphate. The mitochondrial DNA is a very small portions of the DNA and present in the eukaryotic cells, and most of the DNA can be found in the cell nucleus. Also, it is found in the plants and in algae, and also in the some plastid which is generally known as the chloroplast in eukaryotes. Now here you can see the picture in which you can see this is the cell and this is the mitochondria in which the mitochondrial DNA is present. The mitochondrial DNA is the small circular chromosomes found inside the mitochondria, and these organelles found in the cells and called the powerhouse of the cells. The mitochondria and thus mitochondrial DNA are passed almost exclusively from the mother to their offsprings through the egg cell. Now we will discuss the history and the discovery of the mitochondrial DNA. So the mitochondrial DNA was discovered in the 1960 by the group of the scientists. The name was the Margaret M.K. Nass and the Sylvain Nass. They observed the mitochondrial DNA under the electron microscope with the DNA sensitive thread method inside the mitochondria. Apart from this group, another group of the scientists name was the Allen, Hazelburner, Hans Tuppy and the Gottfried Sketch discovered the mitochondrial DNA by the biochemical assays on highly purified mitochondrial fractions. Now, the important features of the organelle genomes. So, here we will talk about some features which are present in the organelle genomes, that is, replicates both in the chloroplast and the mitochondria in a semi-conservative fashion, means the organelle genomes can replicate by the semi-conservative method, and another property is liable to mutations, means they have the property to mutate themselves, means the chloroplast and the mitochondrial DNA both are liable for the mutations. Next property is they are expressed and inherited separately from the nuclear genes, means they are independent from the nuclear genomes and they can be inherited into the offsprings and can expressed. Next property is the all aerobic animals and the plant cells have the organelle genomes, and they are transcribed, can be translated within the organelles. Likewise, the nuclear genomes, as the nuclear genomes have the property to transcriptions and the translations, such as likewise the organelle genomes also can transcribe and translate within the organelles. Another most important feature of the organelle genome is the uniparental inheritance, means it can transmit from the maternal to the offsprings, and this is the uniparental, means in all cases it is observed that the organelle genomes is transferred into the offsprings from the maternal, so the pathway is the uniparental. Another property is it is present in multiple copies in each organelles. For example, the chloroplast DNA may be present up to 20 to 40 copies per chloroplast in a chloroplast, and in case of the mitochondrial DNA, the 4 copies of the mitochondrial DNA may be present, for example, in the yeast. Now, the origin of the mitochondrial DNA, means how mitochondrial DNA evolved. So, the nuclear and the mitochondrial DNA are thought to be of the separate evolutionary in origin, with the mitochondrial DNA being derived from the circular genomes of the bacteria that were engulfed by the early ancestor of the today's eukaryotic cells. This theory is generally known as the endosymbiotic theory, by which we believe that such type of the DNA or the organelle genomes are evolved. In the cells of the extant organisms, the vast majority of the proteins present in the mitochondria, and almost 1500 different types of the protein are present in the mammals, and these are coded by the nuclear DNA, but the genes for some of them are thought to have originally been in bacterial origin. That's why we assume that it evolved from the bacteria. So, having since means the transcriptions in the eukaryotic and eukaryotic nucleus during the evolutions. In this figure, you can see the endosymbiotic theory of origin of the mitochondrial and the chloroplast DNA. The mitochondria and the chloroplast likely evolved from the engulfed prokaryotes, in which you can see that once lived as the independent organisms. At some points, a eukaryotic cells engulfed the aerobic prokaryotes likewise, which then formed an endosymbiotic relationships with the host eukaryote and gradually developing into a mitochondria. Likewise, the eukaryotic cells containing the mitochondria then engulfed a photosynthetic prokaryotes like this, which evolved to become the specialized organelles known as the chloroplast, and like this, and the chloroplast genomes has developed. Now we will talk about the mitochondrial DNA in the different animals, plants, fungi, and the protists. So we will talk first the mitochondrial DNA in the animals. So the animals, specifically bilaterian animals, have a circular mitochondrial genomes. Next is the Medusozoa and Calcarea clades, however, have the species which have the linear mitochondrial DNA instead of the circular mitochondrial genomes. In terms of the base pair, the C. anemone example, the Scharcanthus nocturnus has the largest mitochondrial genomes of any animals, and these are about the 80,923 base pairs long. In February 2020, a jellyfish related parasites name is the Hanigia salmonicola was discovered, and it lacks the mitochondrial genomes. But the jellyfish retains the structure which are the deemed mitochondria related organelles. Moreover, the nuclear DNA genes involved in aerobic respirations and in mitochondrial DNA replications and the transcriptions were either absent or the present, but only as a pseudogenes. This is the first multicellular organism, jellyfish, which known to have this absence of the aerobic respirations and lives completely free of the oxygen dependency. Now the plants and the fungi. So there are the three different mitochondrial genomes types found in the plants and the fungi. The first type is a circular genomes, which has the introns and known as the type 2, and may range from 19 to 1000 kilobase pair in the length. The second genome type, it is a circular genomes, which have around 22,000 kilobase pair, which also has plasmid-like structure, which is 1 kb around, and known as the type 3. The final genome type that can be found in the plant and the fungi, it is a linear genomes, which is made up of the homogeneous DNA molecules and called the type 5. The great variations in the mitochondrial DNA genes content and the size exist among the fungi and the plants, and some plant species also have the enormous mitochondrial genomes with the silene conica mitochondrial DNA, which contain around the 11.3 million base pairs. Surprisingly, even those whose mitochondrial DNAs contain the same number and kinds of the genes which related to the plants with much smaller mitochondrial DNAs. The genomes of the mitochondria of the cucumber, name is the Cucumis sativus, consist the 3 circular chromosomes. The length is around 1556 and 84 and 45 kilobase, which are entirely or largely autonomous with regards to their replications. Now, mitochondrial DNA in the protist. So, protists also contain the most diverse mitochondrial genomes with 5 different types found in this kingdom. The type 2, type 3 and type 5 mentioned in the plant and fungal genomes also exist in some protists. One of these unique type, it is a heterogeneous collections of the circular DNA molecules and represent as the type 4, while the other, it is a heterogeneous collections of linear molecules and known as the type 6. The genome type 4 and the 6 range from 1 to 200 kilobase pair in the size, whereas the smallest mitochondrial genome, which is around which have around the 5,967 base pair, and present in the Plasmodium falciparum parasites. Now we will discuss the replications in the mitochondrial DNA. The mitochondrial DNA is replicated by the DNA polymerase gamma complex, which is composed of 140 kilodalton catalytic DNA polymerase, and this polymerase encoded by the gene which is known as the POLG genes, and two 55 kilodalton accessory subunits which is encoded by the different genes which is known as the POLG2 gene. The replisome machinery is formed by the DNA polymerase, twinkle, and mitochondrial SSB proteins, SSB for the single strand binding proteins. The twinkle, it is a protein and enzyme and functions as unwinds the short stretch of the double-stranded DNA in the 5' to 3' directions, and all these polypeptides are encoded in the nuclear genomes. And here you can see the picture, which is a mitochondrial DNA replications fork structure. You can see here, and this is the oocytes, and this is also oocytes. This is topoisomerase enzymes, and this is the polymerase gamma, and this is the twinkle formations which act as the enzyme like the helicase and unwinds the short stretch of double-stranded DNA from 5' to 3' directions. During the embryogenesis, the replications of mitochondrial DNA is strictly down-regulated from the fertilized oocytes through the pre-implantations of the embryo. The resulting reductions in the per cell copy number of the mitochondrial DNA play an important role in the mitochondrial bottleneck and exploit cell-to-cell variability to the ameliorate the inheritance of damaging mutations. So in this picture you can see the different steps during the replications in case of the organelle genome. Now the genes on the mitochondrial DNA and their transcriptions. The two strands of the human mitochondrial DNA are distinguished as the heavy strand and the light strand, and represented as the H and the L strands. The heavy strand is rich in the guanine and encodes the 12 subunits of the oxidative phosphorylation system, 2 ribosomal RNAs and 12 S and 16S RNAs and the 14 transfer RNAs. The light strands encodes the 1 subunit, that is 8 transfer RNA. So altogether mitochondrial DNA encodes the 2 RNAs, 22 transfer RNA and 30 protein subunits, all of which are involved in the oxidative phosphorylation process. Now you can see the map of the human mitochondrial genome. In this picture you can see the map of the human mitochondrial DNA genomes, which is around 16,569 base pair long, and this is NCBI, National Center for the Biological Information Sequence. Accession number is NC_012920, Anderson et al. 1981, means the sequence is submitted by this scientist in 1981, and the accession number is this, and this accession number belongs to the NCBI sequence number. The H heavy outer circle and the L lighter inner circle strands are given with their corresponding genes in the picture. Now we'll talk about the regulations of the transcriptions in mitochondrial DNA. The promoters for the initiations of the transcriptions of the heavy and the light strands are located in the main non-coding region of the mitochondrial DNA and called the displacement loop or shows as the D loop. There is evidence that the transcriptions of the mitochondria RNAs is regulated by the heavy strand promoter 1 and represented as the HSP1. And the transcriptions of the polycistronic transcripts coding for the protein subunits and are regulated by the HSP2, means the heavy strands promoter 2 regions. The measurements of the level of the mitochondrial DNA encoded RNAs in the bovine tissues and has shown that there are the major differences in the expressions of the mitochondrial RNAs relative to the total tissues RNAs. Among the 12 tissues examined, the highest level of the expressions was observed in the heart, and then it's followed by the brain and then the steroid-genic tissue sample. As demonstrated by the effect of the tropic hormone, that is the ACTH, acetylcholine tropic hormone, on the adrenal cortex cells, the expressions of the mitochondrial genes may be strongly regulated by the external factors, and apparently it's enhanced the synthesis of the mitochondrial proteins, and it's necessary for the productions of the energy. Another interesting thing is that while the expressions of the protein encode the genes was stimulated by the ACTH hormone, the labels of the mitochondrial 16S ribosomal RNAs shows the no significant changes.